Over the next 2 class periods, we're going to go over a little bit more detail of the central dogma of molecular biology, where DNA is transcribed into RNA, which is then translated into protein. So today we'll just talk about what's going on with the DNA and how it codes for protein and also how it gets replicated. So how does DNA encode for information? If you think about it, DNA is just a whole bunch of nucleotides strung together. And those nucleotides are exactly the same except for their nitrogenous bases. And there are only 4 different bases. Bases with A's, T's, G's, and C's. So how do we go from having 4 different nucleotides to having, at least 10 million different species? The key actually lies in the sequence of bases, and it's very similar to how our alphabet works. So our alphabet, we have 26 letters, and we can put those 26 letters together in all sorts of combinations that give us very different meanings. So let's look at these 2 words, master versus stream. Master means something very different than stream, even though they're using the same 6 letters. Now, DNA replicates itself, and when it replicates itself, it has to make an exact copy of itself. Otherwise, when it goes through mitosis or even meiosis, those cells are going to be different, and we don't want the code to be different. So when DNA replicates itself, it's going to do a couple things. First of all, the DNA is going to unzip. So there's an enzyme called DNA helicase, and that enzyme is able to actually unzip or unwind the 2 parallel strands of nucleotides. Next, another protein comes in, another enzyme called DNA polymerase, and it's going to bring in a bunch of new nucleotides here. So once these strands are unzipped, now we have bases just dangling. So for instance, this T here, another A would come in and fill in its spot. So we can see those new nucleotides coming in. There's actually an enzyme that helps guide that process. Finally, once the nitrogenous bases are all paired up, those 2 new strands of DNA will then wind together and form a new double helix. So let's take a look at this really quickly. Here I have a hypothetical series or sequence of bases. So we can figure out what the complementary strand would look like. So A's are always going to pair with the T's. The C's are always going to pair with the G's. So here's our complementary strand. Now DNA helicase comes in and unzips those 2 strands of nucleotides. Now DNA polymerase is going to come in and bring in new nucleotides. So I get a new strand down here, a new strand down up there, and now those 2 strands will wind together, create a double helix, and we have 2 identical strands of DNA that came from just one. So this process is called semi-conservative because we start with 1 DNA strand, it unzips, and we get new parallel strands that come in and form with those base pairs. So only half of each strand is actually made up of new nucleotides. Now, what if there's a mistake in the code? What if DNA polymerase comes in and brings in the wrong nucleotide? What happens? Well, let's take a look at our example here with letters. We have stream, which was an example before. So here's a little stream. Well, if we just switch 1 letter, so here the T, we're going to switch with C, notice how the meaning really changes. So this can happen with our cells. So first of all, how do mutations occur? A little background here. DNA polymerase makes mistakes, and DNA polymerase basically brings in the wrong nucleotide once per 1,000 to 100,000 base pairs. So that might not seem like a lot, but if we think about the hundreds of millions of base pairs that make up our DNA, that actually can be quite a bit. Luckily, we do have enzymes that actually check for mistakes and will fix those before replication is finished. So what we end up with after the proofreading, we only end up with about one mistake every 100 million to 100 billion base pairs. And what that translates to in humans is less than one error per chromosome per replication. So we definitely still get errors in our DNA, just not that many. There are different types of mutations that can occur. So one type is called a point mutation. In a point mutation, there's just a change to one individual nucleotide at a time. So for example, up here, when this DNA strand or sequence got replicated, this adenine got switched to a thymine. And so its complementary base pair got changed as well. So we've just made one change in this sequence. Another thing that can happen is called an insertion mutation. So one or more nucleotide pairs is actually inserted. So if we look at the original sequence of nucleotides here, A, C, T, C, C, T, G, A, etc., we're actually going to insert a whole new nucleotide, and a complementary base pair is going to get stuck in there as well. And so our sequence is the same, except we have a new nucleotide in there, switching things up a little bit. Next thing that can happen is sort of the opposite of the insertion. Instead of inserting a new set of base pairs, one is actually taken out. So here we are missing one of these CG pairs. Finally, an inversion can happen where a piece of DNA actually gets cut off of one chromosome. It might get all turned around and actually reinserted into the gap. So we have the same nucleotides or same base pairs, but now we're going to see their complementary base pairs on either strand. So instead of a bunch of A's and G's up here, now we're going to get mostly T's and C's. So that is also going to change the meaning when it gets translated into a protein. The last thing that can happen is called translocation, where a chunk of DNA is actually removed from one chromosome and gets attached to another. So one chromosome ends up missing a whole bunch of information. Another chromosome ends up with a whole bunch of new nucleotides, so new coding. Now, what's the effect of all these different mutations? We're going to model this next class a little bit, but first of all, there are a couple things that can happen. So oftentimes, mutations are harmful, and they could even be lethal. So depending on the type of mutation, the severity can differ. So lethal just means it's deadly, or it just could cause some sort of disorder in a person or a plant or whatever type of organism is affected. Some actually have no functional effect. So even though there might be a point mutation, maybe it doesn't actually affect the protein that's made in the end, and so it doesn't really matter. Some might even be beneficial. So let's imagine we have a predatory cat. What if there's a mutation that makes the quadricep muscles in this cat a little bit stronger or have a little bit more endurance? That means that cat can run faster and it can run farther. Well, that's not necessarily a bad thing, and we call those types of mutations adaptations, and we'll get much more into that next unit when we talk about evolution.